Dear students, I am Dr. Kumar, Assistant Professor of Zoology, Government First Grade College, Shira. Today's session is the Unit 2, the Phylum Porifera, that is first BSc Zoology students under Tumkur University as prescribed by the university. The Unit 2, Phylum Porifera and Session 5. In this session, we will study the general characters of Phylum Porifera with suitable examples. The learning objective of this session, the students can able to understand the cellular grade of organization, and also they obtain knowledge about the skeletal system and the canal system in case of poriferans. The content of the topic goes like this: the brief introduction about Phylum Porifera and the general characters of Phylum Porifera with suitable examples. Introduction. As all of we know that poriferans are also the primitive invertebrate animals comprising the sponges. Poriferans, sometimes they are commonly called as sponges, and having cellular grade of organization. So in the previous phylum, that is the phylum Protozoa, where you can see the protoplasmic grade of organization. Here, cellular grade of organization, cellular grade of construction can be seen without any tissue or organ formation. Only the cells will perform different vital functions of the body. As the name of a phylum indicates, Porifera, all the members of the poriferans, they are the pore bearing animals. Invariably, the outer surface of the body is having pores, small pores called as osteas. They are very much present. So the body is perforated. Body is perforated means the entire body is having small minute pores, and also the canals, small pores, canals, and the chambers through which the water current flows and passes into the internal canal system, so which is usually lined by the choanocytes and also the pinacocyte. There are two types of cells are present. The outside the body, so they are pinacocytes, and inside the body layer you can see a flagellated specialized cells, they are called as choanocytes. Here you can see the perforated body can be seen, example the cycon and other pore bearing animals, where you can see the body is perforated. General characters. The first character, poriferans are commonly called as sponges. They are multicellular organisms. So whereas the protozoans are unicellular organisms, the entire body is constructed by a single cell in protozoans, whereas here the body is made up of many number of cells, multicellular organisms, which are usually sessile or sedentary in nature. Here, so they are present almost in the same place where they are present, sedentary. Most of them they are marine, present living in the marine environment, and some of them are adapted their life to the freshwater form. Body shape is cylindrical, and symmetry is absent in some poriferans, and in some poriferans the radial symmetry can also be recognized. Body is diploblastic. Di means two, that is outer body layer and the inner body layer. The outer body layer is called as pinacoderm and the inner body layer is called as choanoderm. Respectively, the cells are made up of pinacocytes and choanocytes. The middle layer, in between the outer and the inner layer, there is another layer what is called as mesenchyme, which is always having and it gives space for the different kinds of cells, what is called as amoebocytes. The body wall contains numerous pores. As I told you in the beginning, body is perforated, having small pores called as ostia. through which the water enter into the body through a system of canals, what is called as canal system, into the central body cavity, what is called as spongocoel. The canal system mainly comprises the incurrent canal, excurrent canal, and also the cells, and these canals are lined by a specialized cells, what is called as pinacocytes and choanocytes. Body also contains one or more openings called as oscula. There is a central, a large opening, what is called as osculum, and this osculum open the spongocoel opens to outside by means of an opening, what is called as osculum. Here you can see this slide shows the body organization of the poriferans, where you can see the central body cavity is called as the spongocoel, and it opens to outside by means of an opening called as osculum. This is spongocoel and this is osculum. And if you get into the body wall, here you can see the body wall, where you can see the outer body wall and the inner body wall. In between there are specialized cells called as the amoebocytes. Outer body layer is made up of a specialized cells called as pinacocytes, and the inner body layer is made up of a flagellated cells, what is called as choanocytes. Okay, and here and there you can see the small pores called as ostia. Through this ostia only the water current from outside enters into the body. Okay, and from the spongocoel the water enter outside by means of osculum. The same thing here can be seen here. These are the pinacocytes, these are the choanocytes, this is the ostia, okay, through which the water current enter into the body. Okay, so these are the specialized cells, what is called as the amoebocytes. Sponges feed on minute organisms or small organic particles. Minute organisms or the small organic particles which are present in the water current. Once the water current enters into the body, so these small minute organisms or the organic matter usually catch hold of by a specialized cells called as choanocytes. The respiration takes place by diffusion of oxygen from the water and they flowing into the body. So usually the respiration, the exchange of oxygen and carbon dioxide that takes place between the water and the body. Once the water enters into the body through the ostia or the canal system, the oxygen diffuses into the body and carbon dioxide diffuses out. Sponges have an exoskeleton which is made up of a hard substance, what is called as spongin fibers, or it is calcareous or siliceous spicules, or the combination of both spongin and calcareous bodies can be seen. It is a hard exoskeleton. Excretory product is mainly the ammonia, which is released when the water current enters into the body and it is carried away through the water. Digestion is intracellular. No respiratory, excretory, contractile vacuoles are present in sponges, and in some freshwater sponges all these structures are absent. Reproduction may be asexual or sexual. Nervous system is of primitive type. Neurons are very much present. The nature of neurons are they are bipolar or sometimes multipolar neurons arranged in networks. Bipolar means having two elongated extensions called as axons, and in multipolar neurons, so there are so many cytoplasmic extensions are present. Among these extensions, dendrites, one is the longest, what is called as the axon. Okay, and they are arranged, all these nerve cells, they are arranged in networks. So the entire body is under the network of the neurons. All sponges are hermaphrodites. This is another important characteristic of the sponges or the poriferans. So they have both the sex organs inside the body, both the testis and the ovary, they are present inside the body. Though they are hermaphrodites, cross fertilization is the rule. Okay, though the sex organs are present in the same individual, so the cross fertilization has to occur if the continuation of their progeny takes place. The same thing again, the anatomy, where you can see the pinacocytes, choanocytes, the amoebocytes, and the ostia. Through the ostia, water current enters, and the large central cavity is called as spongiocele. This spongiocele opens outside by means of a small opening called as osculum. Here you can see the asexual reproduction by means of budding. Asexual reproduction, so that takes place either the internal or external budding, or sometimes the formation of the gemmules, the reproductive structures. The sexual reproduction involves the internal fertilization. Cross fertilization is a rule. Male gametes enters sponge body through water current. Male releases the spermatozoa to outside. So once the water carrying spermatozoa enters into the body through the ostia, these spermatozoa are collected by the choanocytes and facilitate the internal fertilization. And in all sponges, the regeneration power is well established. As far as the early development is concerned, cleavage is holoblastic. That means after the fertilization, the diploid zygote is formed, and this zygote, the cleavage occurs when the cleavage furrow bisects the entire zygote into two equal parts passing through animal vegetal central axis. So resulting in the formation of two equal blastomeres, what is called as holoblastic cleavage. The development is indirect, involving a larval stage, free living larval stage, what is called as amphiblastula larva or parenchyma larva. So these are the examples of the phylum Porifera, that is Cycon, can be seen here, Leucosolinea, Spongilla, Euplectella, and some more examples that can be seen. So dear student, this is all about the